Hi Year 10, it's Miss Rasmussen here. This is your week 3 lesson 1 for core and for triple, and we're going to be looking at enzymes in this lesson. So we'll start off as we always do with a recap of the facts. So it's 5 questions for you. What is diffusion? What level of organisation is a nucleus? That's where we put things in levels of organisation. Where would a nucleus be? Which organ absorbs nutrients into the blood? Where is bile made, and which type of transport requires energy? So if you can pause the video, write your answers down and then come back and check. Okay, hi, so welcome back. Checking your answers. Diffusion is the movement of particles from an area of high concentration to an area of lower concentration. The nucleus is an organelle, that is the smallest layer of organisation. The organ which absorbs nutrients into the blood is the small intestine. Bile is made in the liver, and then the type of transport that requires energy is the active one, so it is active transport. So what we're looking at today is enzymes. What we need to know is we need to have a definition of enzymes. So they are biological catalysts which speed up chemical reactions without being used up. We need to know what they are, and you need to be able to explain their active site which their substrate fits into. You need to be able to describe how they can be denatured, which is when the active site changes shape. And then you need to know what can denature enzymes, so high temperature, low pH or high pH. And then finally, you need to be able to state that all enzymes have optimum conditions where they work most effectively. So let's start with this definition for enzymes. Enzymes are biological catalysts which speed up chemical reactions without being used up. So when we talk about biological catalyst, a catalyst is anything which will speed up a reaction. Biological, it's living, it's found in the body. What happens is that it speeds it up, so the enzyme will increase the speed of the reaction. So substances will be broken down quicker, larger substances will be formed together quicker. Chemical reactions tend to think in terms of enzymes, digestion, breaking food down, building molecules up, and then finally not being used up. So at the end of the reaction, this enzyme is unchanged. It isn't involved in the reaction in full. So all it does is it speeds it up, and then you've got an enzyme at the end ready to go again. So how they work. So what you have for an enzyme is you have your enzymes. They're made from proteins, and you have this thing called the active site. We tend to draw it just with quite nice shape. And we've then got the substrate. The substrate is what binds to the active site. If it's breaking it down, it might just be the one and it gets broken down into smaller pieces, but it can also build it back up as well. So you've got your active site and you've got your substrate. The substrate will bind to the enzyme, and this is then called an enzyme substrate complex. Then the reaction happens. So in this case, it's breaking something down. So this substance is broken down, the products come off, and they leave the active site of the enzyme, but this hasn't been changed. So this can be then used again in the next reaction. So it would then come back here, we will get another substrate, and it would catalyse the next reaction. What I would like you to do, there is this sheet in the Teams folder. If not, sketch it out yourself. There are 3 types of enzyme that you need to know about, and these are amylase, protease and lipase. What I would like you to do is to pause the video and then use the video and any additional research that you can find to complete a summary on these 3 types of enzymes. So you need to find out what they are breaking down, where they are made, and any other key information for amylase, protease and lipase. The next bit of the video is going to have some information to help you on this as well. So carbohydrates is the general name. We tend to think about amylase in key stage 3. So carbohydrates will break down carbohydrates, and the one we look at specifically is amylase. And what amylase does is it breaks down starch into sugar or glucose molecules. So you start off with your starch molecule, and this enzyme amylase breaks it down into smaller sugar molecules. It is working in the mouth and the small intestine, and it's made in the salivary glands and the pancreas. Protease breaks protein down into amino acids. So it works in the stomach and in the small intestine. So you've got these protein molecules, and the protease breaks it down into amino acids. And finally, we've got lipase. Lipase breaks down lipids into fatty acids and glycerol molecules. The digestion of fat will occur in the small intestine. So this enzyme is made in the small intestine and in the pancreas. So the difference here is you've got different types of molecules that are formed. So lipase or fats are broken down into fatty acids and glycerol. Now you may have noticed there is a trend with the names. ASE on the end means it's an enzyme, and then whatever we're breaking down goes in front. So lipase breaks down lipids, protease breaks down protein, and so on. So here's just a summary of ideally what you should have. Amylase breaks down carbohydrate or starch into smaller sugar molecules. It is produced in the salivary glands and the pancreas. It will break down starch in the mouth and the small intestine. Protease breaks down proteins into smaller amino acid molecules, and it breaks down protein in the stomach and the small intestine. And then lipase breaks down lipids into fatty acids and glycerol molecules, and that happens in the small intestine. Okay, so the next thing we're going to look at is this idea of denaturing. Enzymes will only work in specific conditions, and they can be denatured. And what denatured means is that the active site changes shape so they no longer work. So here's an example of a denatured enzyme. So if you think back to that model we had where this substrate fits really nicely into this active site, as an enzyme is denatured, it changes and it can no longer fit in. Once this has happened, you can't go back. So if you think about, for example, if you cook an egg is made from protein. Once you've cooked the egg and it's changed white, you can't get this raw egg back. And it's the same with enzymes. Denatured is a really, really important keyword. Make sure that you know that name. They love asking it, and if you don't get denatured, you don't get the mark. So enzymes can be denatured, which means the active site changes shape and they can no longer work. Things that will denature enzymes is high temperature and then low or high pH. Now, all enzymes are going to have optimum conditions, and this is going to be where they work most effectively. And the best way to look at this is to think about a graph. So what we have here is the rate of reaction, and the further up we are, the quicker this reaction is happening. And then we've got our temperature. Now, what we've got here at low temperature, there isn't very much energy, so the particles aren't moving, so there's not much of a reaction. Gets to around 20 and it comes up steeply, but you've got this really small bit here where it is the quickest rate of reaction, and that is what we call the optimum temperature. And then once you pass that optimum, it doesn't take much, it will reduce and eventually stop. And as this goes down, what is happening is that enzyme is being denatured. Exactly the same here for pH. Here the pH, the optimal for this one is just slightly bigger than 7, about 7.5. So again, you've got your reaction, your pH. Here at 3 and at 11, the enzyme is denatured. This 7.5 is the optimum pH. What the optimum condition for an enzyme is will depend on where they are found. So for example, the proteases found in the stomach will have an optimum pH that is acidic because they are working in acidic environments. Lipase will have an optimum pH which is alkaline because they work in the small intestine, which is an alkaline area. So what you need to be able to do for enzymes is you need to be able to define them. So enzymes are biological catalysts which speed up chemical reactions without being used up. They are proteins and they have an active site which a substrate fits into. They can be denatured. This means that the active site changes shape. And then high temperature, low or high pH can denature enzymes. And then all enzymes have optimum conditions where they work most effectively. So make sure that you can learn those facts from the video and that you've got your summary of your 3 digestive enzymes. And I'll see you guys on the next video. Thank you very much.